Today, I'll talk to you about some of our work on deep learning-enabled computational microscopy and sensing. In general, my lab is very much interested in creating better imaging systems, better sensors, better microscopes for use in different settings, field settings, resource-poor countries, etc. So, what you see here on this intro slide are some examples from our previous work where we utilized the smartphone as a platform for microscopy for sensing, all the way from holographic microscopy to fluorescence microscopy to Brightfield microscopy. You can now utilize the smartphone as a platform for conducting advanced measurements, coming close to the measurements images that you capture with benchtop lab-grade counterparts. For example, this is a work from a couple of years ago on converting the smartphone into a dark field fluorescence microscope. With very simple silane chemistry, but very inexpensive sample preparation, we were able to actually see individual DNA molecules imaged with this smartphone-based microscope. These are double-stranded DNA molecules that are labeled with an intercalating dye and beautifully imaged over a large field of using an inexpensive interface. In fact, we can utilize these devices for conducting even more advanced measurements. Here we're looking at targeted DNA sequencing and in situ mutation analysis using the smartphone as a platform, as a reader. This time it's a Brightfield microscope plus 2 different channels of fluorescence, where we were able to look at using an amplification strategy called rolling circle amplification. We were able to understand wild type and mutant at the tissue level using these microscopes inspected with some colon cancer patients to show the utility of these kinds of microscopes for targeted DNA sequencing. So it's come a long way. Maybe 10, 15 years ago, with some of these microscopes, you could barely see a red blood cell, but nowadays it's routine for us to see individual viruses, DNA molecules, count them and size them with these inexpensive interfaces, computational interfaces. On the left, you see here another very exciting platform, which is a computational holographic microscope that's based on-chip holography. If you look at, for example, this device, this is powered by a CMOS imager that we commonly use in our smartphones, 510 megapixel imager. It's at the heart of this handheld microscopy system. The sample goes on top here, and it's a transmissive microscope. We capture holographic images where the sample sits maybe approximately a millimeter away from the CMOS imager. And by changing the illumination from one LED to another, you create super result holograms of the sample, which are then reconstructed to go back to the sample plane, giving you these beautiful images. One of the most important aspects of Lensfree on-chip microscopy, in addition to being compact, cost-effective, and field portable, is its field of view. For example, if you take this image to sample the exact same region of interest with a 40x objective lens, you would have to scan it by a few hundred times and stitch them together. Lens-free microscopy has a very large field of view, and as shown here, this is a tissue section histopathology slide, where if you zoom in, you can see beautiful constructions from these holograms, nicely matching the corresponding image. So these are holographic microscopes, but you can also bring color to these microscopes. As shown here, this is a pneumonia H&E stained lung sample, and by using red, green, and blue LEDs at the same time, together with a Bayer pattern CMOS imager, we can actually create these colorful holographic reconstructions showing the stain of H&E. And the field of view advantage also is highlighted. You can zoom in and show subcellular features. You can continue zooming in and have about easily half a billion to a billion pixels, useful pixels in these kinds of lens-free on-chip microscopy approaches. It's great for spatial telepathology applications, looking at samples, blood smears, pap smears, tissue sections in field settings with inexpensive microscopy solutions, powered by holography, on-chip microscopy, and computational imaging in general. In addition to microscopy, we're also very much interested in utilization of the mobile phone as a sensor. These are some examples from our previous work where we were using it as a sensor, basically, to quantify different types of diagnostic tests, colorimetric, fluorometric diagnostic tests, to look at waterborne pathogens in drinking water, in milk samples, and looking into heavy metal contamination in drinking water. For example, this is a nanoparticle-based assay, which is basically shifting color as mercury ions are within water. And it has a few nanograms per mil level of detection sensitivity on the order of 3 to 4 nanograms per mil, which is more than enough for many applications that demand these kinds of inexpensive field portable sensors. Today, after this brief intro, I'd like to talk to you more about how some of the emerging powerful machine learning tools in specific deep learning has been helping us to create images, to reconstruct images, and to transform images in unique ways, enabling new applications and new imaging science capabilities. So today I'll talk about this unique opportunity that for the last several years, my lab and others in the field have been making some important contributions to. So for image reconstruction, we like using deep neural networks for one very important reason, and I want to emphasize it before I go into the next part of my talk, and that is a deep neural net is a beautiful universal function approximator thanks to all the nonlinear activation functions that it has and all the degrees of freedom that it has. Training a deep neural net for a specific task might take some time and some data, but once it's done, it's a one-time effort and the network is fixed and it implements every time there's a new input in a single feed-forward manner, it outputs it. Computational imaging in general over the last several decades has been utilizing iterative methods to do image reconstruction, image enhancement, and for in general computer vision. But I think one of the biggest exciting features of deep neural nets is the single feed forward reconstruction capability without the need for iterations. And once it's trained, it's fixed. And I think that will be more apparent when I go over some of the next slides, what I mean by this important advantage. So we've been using deep learning for enhancing microscopy at different scales with different modalities. So some of our past work utilized deep neural nets and data-driven approaches to reconstruct holograms. The first holographic image reconstruction and phase reconstruction was done by our lab in 2017 to reconstruct basically holograms with much better contrast without artifacts that are common in standard iterative methods. We also applied deep learning, deep neural nets to take smartphone-based microscopes and improve their resolution and color aberrations by establishing a transformation between the smartphone images and benchtop microscopy images so that the smartphone even gets better in what it can achieve through a neural net. We've also done a lot of work on super resolution, utilizing the smartphone as a platform for beating the diffraction limit of light, using deep neural nets for super resolution microscopy. I'll tell you more about that in the next few slides. And today I'll finish off by sharing with you some of our progress on utilization of deep neural nets and some of these cross-modality transformations for pathology, for virtual staining of biopsy samples. So at the heart of some of the new capabilities that are emerging through deep learning, deep neural nets, what I like as one of the most probably exciting avenues is achieving cross-modality transformations, meaning that one form of microscope, one modality of imaging could be transformed statistically to match another mode of microscopy, another form of microscope. And that enables some unique opportunities that were not possible before, as I'll highlight in the next few slides. So at the heart of these cross-modality image transformations is a training process that typically involves anywhere between a few thousand to a few tens of thousands of registered image pairs, where you have the input images coming from one form of microscopy, and you have the ground truth, that's your wish list that you want to have, and the same samples are imaged by both, and you pair them with beautiful elastic registration algorithms, pair by pair, forming this beautifully registered training image set. With the help of a cost function, loss function that you define, which includes oftentimes a structural loss term and sometimes a discriminated loss coming from a generative adversarial network structure, we train these networks, mostly unit structures, so that we can approximate this transformation that we want to achieve between the input and the output image, matching the ground truth, so that this transformation is accurately learned at different scales by the downsampling and the upsampling of a unit structure. There are different ways of doing this. This is just a generic slide to introduce, but every single application that I'll show will have slightly different cost functions and slightly different network architectures that are fine-tuned to the specific needs of that application. I would say one of the first and most exciting success stories of these types of cross-modality transformations was super resolution imaging. And I'm sharing with you some results, blind testing results of a neural net that was trained to do this transformation between TIRF, total internal reflection fluorescence microscopy, to SIM, structural illumination microscopy. The ground truth here was structural illumination microscopy, where we had 9 different exposures with interfering light to basically create a synthetically enhanced numerical aperture, improving the resolution of the microscopy system at the cost of these structural illumination and multiple exposures on the sample. For example, these are some subcellular features, clathrin-coated structures within a cell that are dynamically assembling and dissolving within the cytoplasm. And at different timestamps, you see the ground truth coming from SIM. If you just don't use SIM and look at a single image with TIRF, this is what you get. These are lower resolution images, and the network is trained for this transformation to do super resolution. At the network's output from a single image on the left, you have a beautiful match to the ground truth coming from SIM, showing you that indeed the network learned that transformation. One of the most exciting aspects of this is you can do this on a single image, input image, as opposed to 9 images that are used for SIM reconstruction. So you have a lower number of images required and potentially less phototoxicity and photobleaching on the sample, which is very important, especially for biological applications. This is a movie of the same thing, except now the cell is live, being captured. It stops every now and then to show you the network's input, network's output, and how it nicely matches the SIM reconstruction performing super resolution. One thing that is quite surprising that we realized is the network actually generalizes very well to different types of samples. We're now taking the same super resolution network that was trained from TIRF to TIRF-SIM. And this network only saw cultured cells during its training. It was trained with just monolayer of cells. Here, out of the blue, we're changing the sample to an embryo, a live Drosophila embryo, which is quite different than a monolayer of cells, obviously, because it's a tissue sample. And if you look at the input images of TIRF, you see substantial deviation in background fluorescence, scattering, all of that. So there was a substantial change in the nature of the sample and how the fluorescence is acquired at the TIRF input. But it's still generalized very well. And if you look at the same network's inference on this blinded testing, it nicely matched the SIM reconstructions of the same Drosophila samples. And this is showing you exactly the same thing, except now we're looking at temporal processes. It's a live Drosophila embryo being imaged, and every now and then we'll stop it and show you the network's input, output, and ground truth comparison, showing you that it indeed generalized to even new types of samples with a network that was never trained with tissue samples in this case. In another example, we have utilized the same UNet structure and with some GAN-based training The transformation this time that we were targeting was confocal microscopy, diffraction limited to STED microscopy, stimulated emission depletion microscopy, which is a nonlinear microscopy modality. What you see here on this slide are blind testing results coming from 20 nanometer particles. So if you look at the ground truth, you can zoom into some regions of interest, and you can see clearly that because of the STED super resolution capability, some of those closed space particles were nicely reconstructed and resolved. If you look at the confocal input at this emission wavelength, they were not resolved. At the network's output, taking those confocal inputs, we were able to resolve them, matching essentially the resolving capability of the STED microscopy system without the spatial illumination of STED microscopy from just a confocal microscope. And if you look at the statistics of this transformation, you can clearly appreciate how the distributions of PSF is matched at the network's output. If you look at the input, this is how those 20 nanometer particles are appearing with full fab maximum of on the order of 300 nanometers. At the network's output, they're left shifted, nicely matching the statistics for the same samples of STED microscopy. So there are numerous other exciting opportunities. This is another one, which is utilizing deep learning to virtually propagate fluorescence images within a volume. We call this framework as DeepC. So it's a very unique framework where you can take a given fluorescence image and define a digital propagation matrix and define a certain three-dimensional surface and virtually propagate this fluorescence image to give you, to yield basically the fluorescence across a certain surface. And this is shown here basically with different DPMs. We were able to take different kinds of aberrations, tilts or cylindrical aberrations as shown here. And at the DeepC output, we were able to basically convert that into a flattened image, taking out those kinds of aberrations by the virtual propagation capability of DeepC. Which means if you were to do longitudinal experiment, for example, this is the elegant sample, it was out of focus for some reason, but virtually now you can take this single video frames and virtually propagate it to any arbitrary plane within a volume to reveal different parts of the sample to be in focus simultaneously without having multiple exposures just from a single image sequence. That's one of the exciting aspects of DeepC. Post-processing wise, you can virtually propagate fluorescence. In addition to fluorescence microscopy, deep learning based cross-modality transformations have been applied also in holography. Holography is a single frequency, a single wavelength, coherent imaging modality. Now we're going to be using these types of ideas to reconstruct holograms with the contrast, with the color contrast, with the incoherence, with the beautiful spatial contrast of Brightfield microscopy, giving best of both worlds. Basically, you take the same three-dimensional samples, you capture holograms of those samples, and those holograms constitute your input images. And at the same time, for the ground truth, you're taking the same 3D samples and scanning them with a high-NA Brightfield microscope to understand how a 3D scanning system would reveal the contrast at different heights. Those are our ground truths, and here a unit structure is being trained to reconstruct those single frequency, single wavelength holograms with the spatial and spectral contrast of Brightfield microscopy. It's an entirely data-driven approach because we do not know the forward model here between single wavelength scattering from objects versus broadband incoherent scattering from objects. So these are some blind testing results on pollen particles. A single hologram is shown here, acquired, encoding the 3D information of the pollen particles. At the network's output, it's reconstructed in a single feed forward through a neural net with the color contrast and the spatial contrast of a beautiful high-NA Brightfield microscope that's scanning the sample. What I show here is the unique ability that this kind of a Brightfield holography network enables us. From a single hologram, shown on the left here, we can virtually backpropagate extremely rapidly digitally to any axial plane within the sample. And at the output of the neural net, we can see, as you're virtually propagating, the Brightfield equivalent image. This could only be acquired with a standard microscope by literally mechanically scanning the volume, and with maybe a few 100 nanometers of scanning step size, capturing that axial volumetric information. So this means you can get volumetric reconstructions equivalent to a Brightfield microscope from a single snapshot hologram. And this is the first time, in fact, that a hologram has been reconstructed with Brightfield contrast and matching not only the spatial features of Brightfield microscopy, but also the color of these pollen particles. Notice that the hologram is acquired with a single wavelength, visible wavelength, and this transformation helped us to also reveal color information of the pollen particles as shown here, which is, I think, one of the very interesting and powerful aspects of these cross-modality transformations when it comes to holographic imaging. These are not just exotic transformations. We utilize them to empower our mobile instrumentation to get better in what we do with them. For example, this is an imaging flow cytometer that is a label-free microscopic system that can image particles as they flow through a system. It has a throughput of on the order of 0.1 to 0.5 liters per hour. As particles are flowing with maybe a centimeter per second speed, it can create beautiful reconstructions of these label-free samples. These are algae particles, plankton that we've sampled in Los Angeles coastal line. Every one of those objects was flowing with about 1 centimeter per second speed through this system. And it's a field portable system that can in real time reconstruct these beautiful images without any chemicals involved. Especially important for looking into algae blooms and predicting those patterns in field samples by having these kinds of field portable instruments measuring continuously or periodically, for example, the oceans. So this is enabled by deep learning based reconstructions, giving you these beautiful contrast and resolution. Another very unique application is looking into mold and pollen spores and bio-aerosols in air. This is a beautiful application of machine learning, both from the perspective of image reconstruction and classification of the reconstructed objects. This device had more than 93% accuracy in classifying different types of mold and pollen particles in air, including black mold. So it could be very useful for example, in the case of a flood, you need to inspect buildings and understand mold growth, etc. This would be a beautiful application where on demand you can sample, detect, and characterize different kinds of bioaerosols. Certainly true for allergens as well. So deep learning enabled holographic microscopy, field portable imaging systems really enable us to look at large sample volumes, air, water, bodily fluids, even in field settings with some unique capabilities. So these are not really exotic transformations that I'm introducing to you. These are very, very useful to empower our field measurement science. I'll finish up in the next couple of minutes with one of the most exciting applications of cross-modality image transformations, which is going to be very, very important for pathology. And this transformation will be from auto-fluorescence of samples, fluorescence microscopy modality at the input, into Brightfield. So two different kinds of modalities. So it's very difficult for us to actually formulate a forward model between fluorescence, autofluorescence, and Brightfield. On top of that, the sample itself, in addition to modality changing, the sample itself will be changing as well to add even more complexity to the system. The sample will be label free at the input, and we want the network not only learn the Brightfield equivalent images, but also of the labeled version of the specimen. So the sample will also go through a chemical process. You may be asking, so what? Why are we looking at this specific and peculiar transformation here? This is actually very exciting for basically histopathology. So every time we take a biopsy, it goes through this laborious process. A biopsy is taken. If you don't stain it with some chromophores, it's like glass. It doesn't have any contrast. For the last 150 years or so, we're staining tissue biopsy tissue with some chromophores so that under a Brightfield microscope, we can see these subcellular features and different contrasts for nuclear features, subcellular constituents of tissue, the connectivity of tissue, fibrin, etc. All of those are visualized by staining. What if actually we could stop this process and take an autofluorescence image of label-free tissue, utilizing the contrast coming from endogenous fluorophores? What if we could actually enable this cross-modality transformation network learn these stained images to be created from autofluorescence input images of label-free native tissue? Then we can actually entirely eliminate the need for technologists, labeling, save time, cost, and make staining a virtual process to be more repeatable. The technology behind this is the same idea of utilizing a deep neural net, but here we're using a GAN structure, which means the generator, which is the virtual staining network, which is also based on a UNet structure, is taking these autofluorescence images, and it's being penalized with some structural loss terms, mean square error and total variation. Every time structurally it's making a mistake with respect to the ground truth, which is the histochemically stained tissue coming from a pathology lab. That's our ground truth. We want to match that. Whenever it makes structural errors, it's being penalized. But on top of that, it's also being penalized whenever the discriminator, another AI, another convolutional neural net, whenever that discriminator fails to understand which one is virtually generated and which one is histochemically generated from a pathology lab, whenever the discriminator fails, or whenever the discriminator understands that the generator created a virtually generated image, then the generator is also being penalized with this discriminator loss. So at the end of this competition between the discriminator and the generator, nobody wins, and we have a beautiful virtual staining network. So I'm sharing with you now some of our images that we've created. Blind testing results. This is a salivary gland stained with H&E. These are coming from histopathology labs, human stained. These are virtually stained coming from DAPI channel of autofluorescence. This is contrast enhanced. What we're using are actually these images, beautifully matching the H&E images of the same salivary gland tissue sections. Here I'm sharing with you Jones silver stain, a spatial stain, which again, we're using DAPI images as input. These are kidney tissue samples. And what you have here is the ground truth coming from histopathology lab. This takes 2 to 3 hours. It's a laborious process to create a Jones silver stain. Now we can do this per whole slide in a few minutes and more consistent than a human histotechnologist. In fact, I'm sharing with you here another type of a spatial stain, Mason's trichome. Liver and lung tissue samples are shown here. This is also a laborious process to create a Mason's trichome. It takes approximately 3 hours where the histotechnologist needs to look under a microscope. We eliminate all of those laborious processes and simplify it to just the acquisition of a label-free autofluorescence image that goes through a virtual staining network, achieving these results. These results are not just virtually identical to the human eye. We've also given these images in a blinded manner to board-certified pathologists, and we asked them to quantify the quality of the stains, nuclear detail, cytoplasmic detail, extracellular fibrosis. Basically, the results confirm that there is statistically no significant difference between our images versus histochemically stained images. They look identical from the perspective of diagnosticians. In another testing, we also gave them these slides that we created virtually versus histochemically prepared, and asked them to diagnose the condition of the patients. These types of tables and comparisons revealed again the same conclusion. There's clinically no significant difference in the diagnosis that they achieve with our images versus standard histochemically stained images. But there is a major advantage of using virtual staining technology, and that is standardization of the stains. If you look at human histotechnologists, This is H&E slides taken from UCLA Health. You can see some variations from run to run, from technician to technician. Whereas at the network's output, these are more consistent. So this consistency is very important for not just human diagnosticians, but for downstream machine learning, tumor margin detection, tumor classification. I think these are also very exciting uses of deep learning and its consistency in virtual staining. Another very, very exciting aspect of virtual staining technology is that from a single cross-section, we can generate a panel of stains that are nanoscopically registered. So you can take one cross-section of label-free tissue, and from that we can generate H&E, Mason's trichome, PAS, Jones-Silver stain that are exactly looking at the same cross-section. Today, or for the last century plus, this was not possible. You would take different sections of tissue and separately stain them to look at a panel. And this would destroy tissue. It's a destructive process. As a result, oftentimes we call the patient back and do another biopsy just so that we can do these types of analysis. This is a huge saving for not only cost, but also it will eliminate unnecessary biopsies. These were examples of label-free to labeled transformation. But you can also do stain-to-stain transformations, which means you can take existing H&E stained images and transform them into spatial stains. Enabled by our stain transformation networks, our label-free virtual staining networks, we can do stain-to-stain transformation using the same framework. In a recent work, recently published last month, we have demonstrated taking, for example, an existing H&E stained biopsy image, this is a needle core biopsy, and transforming it into 3 different spatial stains, Jones, Mason's trichome, and PAS, giving the diagnosticians these 4 panels to show that they can indeed improve their diagnosis compared to having H&E on them. This was very exciting in the sense that rather than waiting for those spatial stains to come to the diagnostician, as soon as H&E comes, the diagnostician can actually make a diagnosis based on H&E plus the spatial stains that are virtually created. This is especially important for organ transplant rejection, where every hour, every day counts, and time savings would be very, very important. So to sum up, to conclude, deep learning-based tissue staining really saves expert labor and time, saves reagent costs, training, personnel cost, and it preserves tissue because we do not deplete tissue. The tissue is preserved. We don't touch it. We don't label it. We don't cut it. As a result, we don't need to have extra biopsies when the tissue is depleted. And most importantly, we can have multiple stains on the same cross-section and have consistency in our staining quality, which would be very, very important. We have a spin-off on this technology. Pictor Labs is commercializing this technology in Los Angeles in collaboration with our team at UCLA. It's a very, very exciting effort, and we're heavily now diving into validation of this technology with experts in different fields to show that this is really the future of staining in histopathology. So deep learning indeed has opened up new avenues for us in microscopy, in enhancing microscopy, in enhancing the resolution, depth of field, contrast, also opening up new reconstruction modules for cross-modality transformations, all the way from coherent microscopy, holographic reconstructions to mobile microscopy, super resolution microscopy, beating the diffraction limit of light, and pathologies I've shown you. Indeed, these are very, very exciting times where we're heavily moving toward thinking microscopy systems that will fuse machine learning with unique hardware to create actually holistically optimized, designed imaging and sensing hardware, which is not the case right now. We have the old hardware augmented with AI to get better. But I think a unique opportunity lies ahead of us where the two work together to design a new microscope of its own kind that is actually powered by AI at the hardware to simplify data acquisition, to reduce unnecessary data, but at the same time perform as good, if not better, in many tasks, including resolution, depth of field, contrast, signal-to-noise ratio, you name it. With this, I'd like to conclude, and thank you very much again for having me here. And I'd like to also acknowledge all of our students, postdocs, undergraduate researchers, and funding agencies behind this work. Thank you.